wanted to start our presentation on an exciting and transformative topic in modern science, the genome editing revolution. The central dogma of molecular biology, DNA to RNA to protein, has been crucial in understanding gene expression. This knowledge laid the groundwork for technologies like CRISPR-Cas9, which enable precise modifications to DNA. So, CRISPR technology, starting with the Cas9 system, revolutionized genome editing across various organisms, including humans. Recent advancements such as BAS and prime editors have made editing even more accurate and versatile, impacting fields from medicine to agriculture.
Talk about in the nucleus. Look at the figure 2.0. We see many types of reduction of DNA. So what is ECORI? ECORI is a reduction enzyme that can cut the double helix. Next, the cleavage domain of fungi can be separate from the DNA recognition domain. Homing in the nucleus are a class highly specific enzyme that their recognition sites are often quite low and unique. So the FN is a function of seeing fungi DNA binding domains with fungi in the nucleus. Tells is gene editing that can bind specific DNA. And also, we have CRSPR combined with various enzyme and charts. This is example. CRSPR. There is one application of CRSPR that is catalytic inactive Cas protein, such as Cas9 and Cas9 to bind target promoters or operating frames. Cas9 is in the nucleus an enzyme that cuts DNAs guided by a guide RNA that contains the sequences complementary to the target DNA. The Cas9 is a variant of the Cas9 enzyme for genome editing and is a variant that has been changed to no longer cut DNA but can bind DNA.
Recombinant DNA technology are two applications of recombinant DNA technology such as functional expression of mammalian peptides and recombinant human insulin production. The discovery of polymerase chain reaction PCR. PCR is founded by Mary Mullis in 1983. The PCR method enabled the exponential amplification of any DNA fragment. There are a lot of variants to the PCR system. For example, PCR is used in rational mutagenesis using site-directed mutagenesis method. Second, PCR is also used in random mutagenesis using error-prone PCR. These techniques are instrumental in generating genetic libraries for laboratory evolution.
Scientists have figured out the complete DNA sequences of many organisms, starting with simple ones like viruses and bacteria, and moving on to more complex ones like yeast, plant, and human. This was initially done using a technique called Sanger sequencing. The article explained three main ways DNA can recombine or integrate into another DNA sequence. The first one is homologous recombination, a process where similar DNA sequences exchange parts, often used for DNA repair. The second one is site-specific recombination, a process where DNA is inserted into a specific spot on a chromosome, often used by viruses to insert their DNA into a host. The third one is recombinering, a method to introduce specific changes to DNA in bacteria by inserting synthetic DNA sequences. In summary, this passage discussed the history of sequencing and editing genome, detailing how different methods and tools have been developed to manipulate DNA for research and potential therapeutic purposes.
Argonate proteins and CRISPR-Cas systems are two key tools for targeting and modifying genetic material. Argonate proteins AGO are involved in RNA interference, a natural process in eukaryotic cells. They use small RNA guides to find and bind to missing mRNA, either silencing a gene or cutting the mRNA. This has been used to treat some genetic diseases. There are also prokaryotic versions of argonate proteins that target DNA, but they are less efficient than CRISPR at editing double-stranded DNA. On the other hand, CRISPR-Cas systems also use guide RNA to target specific DNA sequences, with Cas9 being the most famous for genome editing. CRISPR systems can make precise cuts in DNA, but they need a specific sequence called PAM to work efficiently. While argonate and CRISPR both use nucleic acid to target genes, CRISPR is generally more efficient, especially for precise DNA edits.
In conclusion, the article ties together this result by discussing the implication for real life. The development of genome editing tools has not only advanced our fundamental understanding of biology, but also led to societally relevant application. For instance, CRISPR-Cas technology is being used in medical research, agriculture, and even more.